Yeah, I wanted to thank the organizers for allowing me to give this presentation. It's a great honor to be sharing the stage with the other illustrious speakers. I wanted to tell you that Dr. Ribeiro actually allowed me, since I'm not a pathologist, to change the title of the talk somewhat. It is now Current and Emergent Molecular Targets in Lung Cancer Therapy. But I do promise you I have one slide about the cytopathology aspect of this and how tissue should be managed, because that really is one of the challenges as well. So I do not have any financial conflict of interests, but since we are also talking about some of the emergent molecular targets, I have to disclose that not all compounds have an FDA approval for the discussed indication. So the definition of molecular targeted therapy, when you define a target, you have to think about two separate things. So targeted therapy is defined as a type of treatment that blocks a specific pathway that's required for tumor cell survival, rather than what we traditionally thought about chemotherapy as merely interfering with rapidly dividing cells. And where this definition of the target becomes important is in that you have to decide on whether or not the tumor cell survival absolutely depends on one pathway that's altered, or whether or not this is one of many pathways that's sort of contributing to tumor cell survival. Now, why is this important? About 10 years or so ago, we discovered that the concept of oncogene addiction, which essentially means that a cancer cell is absolutely dependent on the presence of one single mutation, to be true in lung cancer as well. This is something that we knew for a long time in CML, where the BCR-ABL overexpression of the transgene of BCR-ABL has long been known to be sufficient for tumor cell survival. But with the development of the EGFR inhibitors, and more important, the subsequent identification of EGFR activating mutations, we got to know that the concept of oncogene addiction is true in lung cancer as well. This CAT scan is from the original paper from Thomas Lynch in the New England Journal. This is a patient with an EGFR mutation. When you read the paper, you will find that this was a pretty sick person already. This was not given in the first line, and within a couple of weeks, you saw a dramatic improvement in the radiographic findings of the cancer, and even a faster improvement in symptoms. So my objectives for this presentation, also in the interest of time, is to discuss established targets for which FDA-approved therapies exist. I want to discuss some emergent targets that are currently under clinical evaluation. And I want to discuss some strategies that are also in clinical evaluation to overcome resistance to established targeted therapies. I will not, again, in the interest of time, talk about biomarkers that sort of predict response, heightened response to therapies. You may be familiar with ERCC1, RRM1. Those are biomarkers that predict response and resistance to platinum-type drugs and gemcitabine. But the recent clinical trials that assign patients to specific chemotherapy arms based on the expression of those biomarkers have been negative, and hence I will not discuss this in much detail. So when our employer still had money to spend on stimulating the economy, a lung cancer mutational consortium was founded as part of the stimulus package. And the intention of this was to determine in patients with adenocarcinoma the frequency of 12 previously described driver mutations. And not only that, but to also develop clinical trials to target these driver mutations. A driver mutation, again, is thought to be a mutation that's capable of inducing a malignant transformation. So tumors with driver mutations are thought to be tumors that are oncogen dependent. Currently, there are 14 centers around the country. The goal is to analyze the frequency of these driver mutations in about 1,000 patients. And at this year's ASCO, the sort of preliminary results of the study have been presented. So 1,100 patients had been enrolled. 1,000 patients had been tested. 733 of those 1,000 patients had enough material to be tested for all 10 genes. There were 60% women in there and 30% never smokers. So you will see that this pie chart that I showed you and that I will show you again in the next couple of slides may not be entirely representative for our population in the VA, where the smoking rates in the gender distribution is really quite different, and the rates of non-smokers is really quite a bit lower. But what you see here, and this is, I think, very interesting, that overall survival does track by the presence of a mutation and whether or not a patient received targeted therapies. These are not the final data yet, but patients who had a driver mutation and who received targeted therapy had an overall survival of, a median overall survival of 3.5 years, whereas patients who did have a driver mutation and did not receive targeted therapy had an overall survival that was about a year less. And patients without a driver and patients who did not receive targeted therapy had a survival that was about the same. So you're all familiar with EGFR. EGFR is sort of the primary oncogenic pathway in non-small cell lung cancer. EGFR inhibitors, such as gefitinib and erlotinib, were actually developed based on the fact that there's a high rate of overexpression of EGFR in lung cancer. About 50 to 60% of all lung cancers overexpress EGFR, but mutations are much less common. And only occur in about 10 to 20% of patients with adenocarcinomas. They're virtually non-occurring in patients with squamous cell carcinomas. Downstream from EGFR is, and I'm sorry, this projects not tremendously well, is RAS, so that would be in this blue circle here. And downstream from RAS is BRAF, MEK, and ERK. Over here is AKT and mTOR. We've long known that KRAS is an oncogenic driver as well, and this has been shown both in mouse models as well as in humans. We do know that all of these oncogenic driver mutations are virtually exclusive. So it's very rare to have an EGFR mutation and a KRAS mutation in the same patient, for example. So the role of EGFR inhibitors in the first-line treatment of non-small cell lung cancer is very well established. It's probably one of the best established principles in the treatment of non-small cell lung cancer, period. And there are a number of studies that show this. The first one was the IPAS study, where a subgroup analysis was done. The IPAS study was initially planned as a non-inferiority studies for patients that have a high clinical likelihood of being EGFR mutated. So this was a study that was done in Asia. They enrolled predominantly women, people with very low smoking status, and all of these patients were of Asian descent. What they wanted to show was that gefitinib is as good as chemotherapy with carboplatin and Taxol. It turned out it was as good, so the primary endpoint, the non-inferiority, was met. But when people were separated, patients were separated by the subgroups of EGFR mutated tumors versus EGFR wild-type tumors, it turned out that the EGFR mutated tumors had a significantly prolonged progression-free survival if they received gefitinib, whereas the EGFR wild-type tumors did significantly better if they received chemotherapy. And in subsequent studies that were both done in Caucasian and in Asian patients, both done with gefitinib, erlotinib, and most recently afitinib, which is an irreversible inhibitor of EGFR, those results were confirmed. So the second target for which we have an FDA-approved drug is the anaplastic lymphoma kinase, ALK. This is not a mutation, but these are tumors that have a transgene between EML and ALK, which leads to overexpression of the gene. This transgene in mice is capable of inducing tumors as well, meeting the definition of oncogene addiction again. We had the crizotinib, the ALK inhibitor, actually received FDA approval a couple years ago based on the results of the phase 2 study. The FDA required the company Pfizer, though, to conduct a randomized phase 3 trial, which was published this year in the New England Journal. A one-to-one randomization to crizotinib versus chemotherapy with either pemetrexed or docetaxel. The rate of the translocation was a little bit less than 10%. They had to screen 5,000 patients to enroll 347. The primary endpoint, like in all these studies, is progression-free survival. Secondary endpoints were response rates, overall survival, toxicity, and importantly, patient-reported outcomes. So what you see here is that the progression-free survival with crizotinib is significantly superior to the survival with chemotherapy, and it appears that pemetrexed is slightly superior to docetaxel in these patients. So these are all patients with metastatic disease. Objective response rates, about 65% versus 20%, and those are response rates that are very comparable to the response rate that you have seen with gefitinib or erlotinib in EGFR mutated tumors. Crizotinib is quite well tolerated. The major toxicity that you would have to watch out for are LFT abnormalities, which happen in about 40% of the patients. I think this is an important slide. In the beginning, when we sort of introduced mutational analysis to our patients, the fellows in the clinic would often ask, why do we have to do this? We could either start with chemo and then follow up with targeted therapy versus targeted therapy that you can only follow up with chemotherapy. But I think the patient-reported outcomes are something that's very important. What you see here is that virtually all parameters point towards improvement in patient-reported outcomes with crizotinib, whereas most of them point in the opposite direction with chemotherapy. So if you have a symptomatic patient, if they go on targeted therapy, they're much more likely to improve clinically than if you were to put them on chemotherapy. This has not been done with the EGFR mutated patients, but what we see clinically is exactly that. So I promise you to come back to this pie chart one time. It's not only important to know about mutations, it's also important to be able to target these. And while currently we only have 2 drugs with FDA approval, such as erlotinib and crizotinib for mutations, you see here on the right side of the table that there are multiple different drugs in clinical development, and I will show you in the next couple of slides some quite promising data for some of these compounds. So go back to this pie chart one more time. You see that obviously the highest potential yield and the highest potential benefit could be achieved in patients that are KRAS mutated. Now, we've been trying to target KRAS for the last 20 years since we know that KRAS mutations are fairly common. You may remember the sort of initial excitement about farnesyltransferase inhibitors about 10 years ago that didn't really pan out to be beneficial. the current strategy is it's very difficult to develop a small molecule that's targeting the KRAS mutation itself. So the current strategies focus on targeting downstream effectors of KRAS, and the most promising so far is the MEK tyrosine kinase, and I will show this in a second. So this is a phase 2 trial that was presented at ASCO the previous year and has subsequently been published in Lancet Oncology. And I'm sorry, the slide didn't fit exactly. But the approach here is to combine the MEK inhibitor selumetinib with docetaxel in the second-line setting versus chemotherapy, standard chemotherapy with docetaxel in patients with metastatic non-small cell lung cancer, which were KRAS mutant. The primary endpoint in the study is overall survival. Secondary endpoints were progression-free survival and objective response rates. And what you see here is that this is actually a negative trial in terms of the overall survival endpoint, although there's quite a big difference still between 9.4 versus 5.6 months favoring the selumetinib arm, but the p-value is not statistically significant. Relative response rates, 37 versus 0%. 0 is probably a little bit lower than what you would expect with docetaxel, but not that much. In the second line trials, the response with docetaxel was about 5 to 8%. And what turned out to be significantly different in this trial was the progression-free survival between 5.3 and 2.1 months. So even though this study was negative, the difference in progression-free survival for the company that's supporting this trial justifies to take this to phase 3, and the phase 3 trial, to my understanding, is on the way. So Dr. Lawson had just talked about the BRAF inhibitors, and you see in my pie chart that there are some lung cancers that are BRAF mutated as well. The frequency of BRAF mutations, the V600E mutations, is much less common in lung cancer than in melanoma. It's about only 2% of all adenocarcinomas that are BRAF mutated. At ASCO this year, some interim results of a phase 2 study with dabrafenib, which is another V600E specific BRAF inhibitor, were presented. 17 patients so far were enrolled. This is a multi-center trial. 12 patients of those were evaluable for response. There were 7 partial responses and 4 progressive disease, 1 patients with stable disease. So if you remember the 66% of people with response that we had seen in the crizotinib trial that we see in the EGFR inhibitor trial, it's sort of coming back to that same number in BRAF mutated patients as well. The duration of response in patients with the duration of response in patients that had achieved a response is between 30 and 49 weeks, and this may be a little bit longer than what's been seen in melanoma. And the toxicities are in line with known toxicities of these drugs. This is a publication that was in Cancer Discovery. An even smaller piece of the pie are the RET fusions. This is a study that's undergoing at Memorial Sloan Kettering. They published this paper with 3 patients enrolled. They had 2 responses as well so far, so it's coming down again to the 66%. But it will obviously take quite a bit more time in the expansion to other centers to be able to complete this trial. Now, EGFR inhibitors have been in clinical practice for about 10 years now. Erlotinib has been approved 10 years ago. We have been treating patients with EGFR mutations for about this period of time, so we have a fairly good understanding what's happening molecularly if these patients relapse, and everybody does relapse. None of these targeted therapies are curative. And what you see here is that the biggest piece of the pie, about 45% of patients develop a second EGFR mutation, the so-called T790M mutation, that renders the primary tumors resistant to the effects of EGFR. The MET amplification had received quite a bit of attention because that was the second mechanism of resistance that had been described, but it's actually relatively rare. It's only about 5% of tumors. A larger percentage of tumors actually has transformation into small cell lung cancer, and the right treatment for those patients is a small cell regimen. If you get the small cell component back into remission, these patients may actually continue to respond to EGFR tyrosine kinase inhibitors. Based on the high frequency of the T790M mutations, there was an intense interest in developing irreversible EGFR inhibitors that are active against the T790M mutation. The two sort of first generation irreversible EGFR inhibitors, afatinib, which has now received FDA approval for EGFR mutated tumors in the first line, and neratinib were both tested in this setting, but results were very disappointing. And the feeling is that these these drugs are actually not specific for the T790M mutation. You have to push it up. If you want to target T790M, you have to push it up into a range where you target wild-type EGFR to quite a significant extent and are sort of limited by toxicities. So the feeling is that these trials were negative because it was not possible to achieve sort of selective inhibition of the T790M mutation. This is about to change possibly. A EGFR selective, a T790M selective EGFR inhibitor has been developed. It's in phase 1 clinical trial now. There were sort of lots of intellectual property issues initially that prevented this drug from going into clinical trial initially, but those seem to have been worked out, and this compound is available at Dana-Farber in phase 1 trial. Our understanding of the resistance mechanisms to ALK translocation is lagging behind a tiny little bit. There was a study in Translational Medicine and in the New England Journal earlier this year, which shows that also in the ALK kinase, there is an emergence of mutations and amplifications. Some of these tumors have c-KIT amplifications as resistance mechanisms. So this is the slide that I promised you, and I hope you can read it. The problem, or the emerging picture here, is that these are all patients with metastatic disease. So these patients have only small biopsies available that are either derived from bronchoscopic specimens or from CT-guided specimens. So prioritizing your samples for biomarker analysis is something that really becomes quite important. What Dr. Travis suggested in this paper was to limit the use of immunohistochemistry to determine the histologic subtype, adeno versus squamous cell carcinoma, to one marker each. In those tumors where a clear-cut diagnosis has not been possible to be achieved, and to basically save the rest of the tissue specimen for molecular analyses. So what's happening in earlier stages, and this is going to be very quick. We still don't know, despite the fact that these drugs have been available for so long. The first study that looked at adjuvant gefitinib, the BR19 trial, was terminated early once gefitinib lost its FDA approval, its provisional FDA approval. But the study was negative. There was no benefit to adjuvant gefitinib. And interestingly, that benefit was also not seen in patients that had an EGFR mutation. This was presented at ASCO a number of years ago and has just been in the last JCO. The RADIANT study is a very similar study design for Tarceva. That study has completed accrual, but has not been published yet or presented yet. There are about close to 1,000 patients in this study. Now, the problem is that also not EGFR mutation was the primary inclusion criteria, but EGFR overexpression by either immunohistochemistry or FISH. There are about 20% of patients with adenocarcinoma that are EGFR mutated, but whether or not that number is large enough to tell whether or not there's a benefit from Tarceva in EGFR mutated patients, we don't know. And this brings us to the last slide. The future direction that the NCI wants to go to is the Alchemist initiative, adjuvant lung cancer enrichment marker identification and sequencing trial. What they're proposing is to test patients with resected lung cancer for EGFR and ALK translocations and enroll them into biomarker-driven trials. 4512 through ECOG with olatinib and the A081105 trial with crizotinib, and to use the tissue from all the other patients for the TCGA project. So the conclusions are olatinib and crizotinib are very effective therapies in oncogene-addicted non-small cell lung cancers. Novel targets and targeted therapies are rapidly evolving. High-quality biopsies for molecular testing are becoming key, and it will become even more important as time goes by. Don't waste too much tissue for immunohistochemistry. Discuss the molecular markers that you need with your pathologist. I think the need for rebiopsies in patients that were treated with a targeted agent to determine the resistance mechanism is emerging. And I thank you for your attention. Sorry for running over a minute.
Okay, thank you for that very excellent talk.